Good morning, everyone, and welcome to today's primer. This is our second to last primer of the season, and I'm really excited today for this topic of long-read sequencing. Today, our speaker is Kiran Garimella. He is the Director of Computational Biology in the Data Sciences platform at the Broad, where he is also an Institute Scientist. Kiran has extensive experience in the development and application of cloud-based data processing methods and automated analyses for third-generation sequencing data. He currently leads the Data Sciences Platform's long-reads group, which aims to transform the genomics landscape by undertaking long-read sequencing of human and infectious disease genomes. Kiran earned a DPhil in genomic medicine and statistics from the University of Oxford Wellcome Trust Center for Human Genetics and completed postdoctoral studies at the Oxford Big Data Institute. He is now currently co-chairing the NIH's All of Us Long Reads Working Group, which is undertaking the discovery and analysis of previously unseen variation in 20,000 diverse individuals. And today he'll be talking to us about sequencing and analysis of long-read whole genome data. Thank you.
Wonderful. Thank you for that introduction. Thank you for all for joining us for this session. Let me set the stage a little bit. I think in most of the sequencing primers, we're largely talking about the Illumina instrument. I think Brendan Blumenstahl was here just a few months ago talking about all of the platforms that are supported by the Genomics platform at the moment. I'm going to talk about the platforms in the middle and the right of the screen. The instrument from Pacific Biosciences, or Bio, and the instruments from Oxford Nanopore, or Nanopore. Both long-read instruments. Just quickly, why do we care about long-read data versus what is, I think, the workhorse of the genomics field right now, which is the short-read Illumina instruments? Well, in recent years, we've seen, I think, the same story recapitulated in a few different places, that structural variant discovery with the long-read platforms is just vastly more accurate, more comprehensive than what we get with short-read data. The top panel is a really nice figure from Ebert et al., just comparing the rate of structural variant discovery per sample in short-read data to that in long-read data. So you can see the short-read data is discovering around 10,000 SVs per genome. Now, the nature of short-read data being short reads around 150 bases is that there are a lot of places in the genome where the short-read data simply can't traverse. Largely repetitive or highly diverse regions. Ironically, the loci like that are substrate for structural variant formation. So the fact is the short-read data just can't get access to the loci where a lot of these structural variants live. When we compare this to long-read data, which typically has 10 to 20,000 base reads, we're seeing around 25,000 structural variants per genome, 25 to 30,000, depending on a number of different factors, what reference sequence you use, what population background. But that's what we're starting to converge on those numbers as the typical number of structural variants that we see per genome. On the bottom, you can just see the length distributions, and you can see the red lines, which are Illumina sequencing, and orange and gold, sort of fall off very quickly as a function of length of the event, whereas the long-read sequencing is much better able to capture those longer events, right? So we think that in a short-read genome, we're missing something like 50 to 75% of the structural variants that are there. And these events are important, right? We're starting to really be able to dissect the contribution to complex traits and quantitative loci through structural variants. This is a figure from Cheng et al. Showing, this is actually a short read analysis, but showing very large structural variants in association with massive changes in gene expression, right? Again, structural variants are largely falling in these repetitive regions, upstream from genes in promoter regions, have an outsized effect on the regulation of expression of those genes. Or I think one of my personal favorites, this is from a really nice explainer by Vera Rajagopal, who I think is at Regeneron, talking about a 25-year-old mystery that is the causal variant for spinocerebellar ataxia type 4. In 1996, Flanagan et al. Showed that in a 5-generation pedigree, through linkage analysis, that the causal locus was somewhere in the 16q22.1 region of the genome. This is a region that we now know is very GC rich, has a huge amount of segmental duplication activity, just a locus that is more or less completely intractable with short reads. 25 years later, with the advent of long read sequencing, we can see what the driving variant there was. It's a repeat expansion, just shown on the right, a GGC repeat expansion. The repeat expansion is pathogenic when it starts to hit more than 40 units. You can see in red, there are a number of individuals in their study that were at 45, 50, 60, 70 units. So capable of seeing this locus now with long read data, but just for a moment, just think about the numbers, right? The triplet repeat, 3 bases, 60 or 70 units of that. That's only 180, maybe 200 bases, right? So just beyond the reach of short reads, but with 10 to 20,000 base reads, this is not a difficult locus to characterize. So really interesting stuff that we can now start to access these compartments of the genome that have previously invisible to us. So again, let's put the two side by side. Short reads, long reads. Short reads around 150 bases. We align them to a reference sequence. We see a bunch of gaps in our analysis where those reads can't traverse because of some partial homology to some other part of the genome. With long reads, we are able to map those reads uniquely. We're able to characterize large variants, large repeat expansions in just one go. And that makes a lot of problems that were previously very difficult, like very, very tractable. Genome assembly is a prime example of this, right? It's difficult if you're going to take a genome and carve it into very small 150, 200 base fragments. It's akin to solving a puzzle with a lot of very small pieces, many of which look very similar to one another. How can you know what the right orientation, right position is for any of those pieces? Long reads is just larger pieces, right? It's much easier to figure out how everything fits together. We see this manifest in the data. Here's a really nice example from one of my colleagues, Fritz Sedlacek at Baylor College of Medicine. And Illumina data is at the top. And our two long read platforms, PacBio and Nanopore, are the middle and bottom tracks respectively. You can see this deletion of about 100 and I think it's 100 bases or so, 115 bases. What you're seeing in the Illumina data is just an absence of coverage. You're not seeing reads really traversing. The deletion is just too large. Whereas in the PacBio and Oxford Nanopore data, you just read straight through very cleanly. You see, you see that deletion on the right-hand side. The insertion, which is I can't tell from this image, but a couple hundred bases long. Again, you see some evidence that that IGV is giving us in these multicolored reads. It's indicator that something has not mapped properly. But the actual insertion sequence is not evident in any of these reads. Whereas in the PacBio and Oxford Nanopore reads, you see that signal clear as day. Right. So we're seeing these larger events that we haven't been able to see before, but also, and I'll get into this in a moment, long read data from both PacBio and Nanopore is 2 experiments in one. Right. So we're seeing primary genome sequence data. We also see 5-MC methylation data. Thankfully, the visualization tools that I think we all know and love have been updated to show us this information. So here, what I'm showing is the 5MC signal that came off of the instrument. This is achieved through direct readout of the sequencing data. There's no bisulfite prep or anything like that. It's just inherent in the data, and I'll talk a little bit about how exactly that signal is derived from this data. But the blue regions are regions of low methylation, red high methylation. You can start to see just the methylation pattern across this really interesting locus. That methylation data is quite accurate when we compare to a few different things. So on top is comparison to traditional bisulfite treatment of short read sequencing. You can see that accuracy varies as a function of coverage, but it's around 20-25x. We're seeing 90 to 95% correlation with what we see from bisulfite sequencing. If we compare both the Oxford Nanopore and PacBio platforms, they are largely in agreement as well. Again, this varies as a function of coverage, but the two instruments are correlated to close to 95% as well at in their characterization of methylation. Okay, let's take a step back. How do long-read sequencers work? I put the Illumina sequencer here for comparison. I find that people generally know this best, so I'm going to start with that, and then we'll talk about PacBio and Nanopore. So just a reminder, how does Illumina sequencing work? Well, we start with fragments that we attach the Illumina adapters to, which then attach to a flow cell. We do bridge amplification on the flow cell. Each amplification step forms a cluster of identical templates in some region of the flow cell, to which we start flowing in complementary and fluorescently tagged nucleotides, building up that sequence one base at a time, imaging every step of the way. Right, so the sequencing here is by synthesis. We're using the fluorescently tagged nucleotides to report exactly what nucleotide is present at at a given position, and the superposition of that fluorophore activity in a colony across a lot of different templates is a strong enough signal that it can be imaged by the onboard camera. There are some similarities with PacBio sequencing in terms of its exploitation of complementary bases that have been fluorescently tagged, but the architecture is vastly different, right? We're not sequencing a colony of identical molecules anymore. This is single molecule sequencing. So now we are imaging a single DNA fragment rather than a cluster of DNA fragments. And then the way we're doing this is there's an initial prep to attach what PacBio calls these SmartBell adapters, these hairpin adapters that basically take a linear DNA template and circularizes it. Each one of those templates goes into a well that is called a zero mode waveguide. Each well, it has so the DNA fragments are Poisson distributed across these wells. Some of the wells have exactly one read, which is good because that means that the signal that we're getting from that well is not corrupted by other reads that are simultaneously being sequenced and generating signal that is very difficult for the base caller to disentangle. Some of the wells have zero reads. You don't get any signal at all. So even though there are a huge number of wells on the flow cell, not every single one of them produces valid signal. The software on board the instrument is responsible for figuring out which wells produce valid signal and don't. So we don't have to worry about that part. But just to say that despite the fact that there are 25 million wells on this flow cell, you don't literally get 25 million reads out of it. You get maybe a quarter of that, half of that, something like that. So, just imagine for valid sequencing, only the wells that have a single read in it are the ones that we care about. And then, the polymerase, which has been annealed to the open part of one of these smart bells, will start synthesizing the complementary base. That enzyme has strand displacement activity. So as it sequences the complementary base, it's actually unzipping the DNA so that it can start adding more and more bases. And so out of this circular construct, you get a linear sequence that is that DNA redundantly sequenced a number of times. The readout of this is a signal intensity over time, right? It's not individual pictures like Illumina, where they are able to arrest and restart the sequencing process with every nucleotide added. Instead, it's a real-time readout. And so the base calling software on board the instrument is reading these, what are quite literally termed movies, and turning that into a base calling signal. Now that is quite error prone still, right? It's a fairly noisy signal that you're getting from one read rather than a cluster of reads. So it's pretty high error rate, around 10 to 15% errors. The dominant error mode is insertions and deletions, right? Because if you look at the signal, it can be very difficult to tell, if I have a sustained, homopolymer A tract, while I'm seeing a movie, the polymerase is, not necessarily incorporating bases at a constant rate. So it can be quite difficult to tell, am I seeing, 10 A's, 11 A's, 15 A's? And so there are still some errors that accumulate in this sequence. This is where the redundant sequencing of PacBio really shines, right? So like I said, we are taking a linear DNA template, annealing these hairpin adapters to it, and then redundantly sequencing around this circle. Generally, we see something like 10 to 30 passes around this circle. That is 10 to 30 copies in both forward and reverse directions that we can use to compute a consensus sequence. So after the consensus sequencing generation, we go from an error rate that is around 10 to 15% to something way less than that. I think we generally, less than 1% these days, less than 0.1%. I think, these days we talk about PacBio sequencing with an accuracy of around somewhere between Q30 and Q40. So comparable now to Illumina sequencing, which was not true a number of years ago. Like I said, there are 25 million reaction wells on the flow cell. This is actually up from the previous iteration of the instrument. So the new Revio instrument, which just came out 2 years ago, has 25 million wells, will generate data in around 24 hours. Each one of these flow cells generally produces a 30x genome. I think we generally say 24 to 30x. Now, whoops, I think that's, I have these slides out of order. I'll skip around. So nanopore sequencing, how does this work? Now, interestingly, the Illumina sequencer and the PacBio sequencer are all essentially hijacking polymerase to do a lot of its work. The nanopore instrument is quite different, right? There is no polymerase involved. Instead, it's a completely electrical readout, which is why that instrument is much smaller, right? There's no imaging platform. It's instead a voltage sensing platform. So in nanopore sequencing, there is a membrane in which an engineered, I think a mostly engineered pore resides in that membrane. A single strand of DNA is translocated through that pore, and each nucleotide essentially it's kind of it's a resistor. So there's a voltage across the membrane, and as the nucleotide flows through, it changes the characteristic current that you are observing. And so it's a real-time readout, but rather than optical signal, what you're seeing is electrical signal. There's a combination of things that Nanopore does in order to ensure the accuracy of their data, right? So Nanopore relies on deep neural networks in order to characterize and base call signal trained off of a huge amount of yeast data, human data. With the advent of the telomere to telomere data, I think there's a lot of really high quality human data that is now available for training the base caller. And Nanopore has a bunch of additional tricks that they are starting to employ in order to more accurately characterize their reads. Right. So unlike PacBio, which can do this circular consensus sequencing trick, Nanopore can't really do that circularization and sequencing. Instead, what they do is, I think by default they do they do what they call simplex sequencing, which is just sequencing a single strand of DNA. Now what they're starting to do is attach some adapters that promote the instrument sequencing the second strand immediately after sequencing the first strand in the same pore. And they informatically pair the forward strand and the reverse strand in order to compute a consensus sequence for those two copies of the same read. So you could think about it. You could think about it as similar to circular consensus sequencing, like you do in PacBio, except rather than having 10, 15, 20 passes on the molecule, you have 2. The rates of this are increasing. When they first introduced this a few years ago, maybe only 20 to 30% were actually sequenced in this duplex mode. Now I think it's pushing 70 to 80%, and Nanopore continues to push and try to get this as close to 100% as possible. There are additional steps that Nanopore is starting to take. So similar to PacBio, having a number of machine learning tricks up its sleeve to better characterize the signal from their instrument. I think Nanopore relies more on the community to come up with innovations like this. This is one innovation that has just recently been preprinted just a couple of months ago. A tool called Hero, which is a deep learning tool for error correcting Nanopore reads that are at least 10 kb in length. They have this really nice convolutional neural network model for learning the manner in which the Nanopore instrument makes errors and error correcting those sequences. And that the application of those tools pushes the quality of the data from around Q20 to above Q30, quite often around Q40. You can see in the plots on the right-hand side just the quality score before and after the application of some of these error correction tools. Read length distribution. So like I said, so PacBio it has optimized their process for error correction. As a result, they keep their read length distributions quite tight, around 15 to 20,000 bases. That seems to be the sweet spot to get a maximum number of consensus passes around the fragments that they're sequencing. So they're ideally always trying to see at least 5 to 10 copies of every single read. Nanopore is much more versatile in this because it doesn't have that circular consensus process. It doesn't have as strict a read length distribution requirement. So typically what we see is generating 10 to 20 kb libraries for the instrument, but Nanopore instruments can also be run in what's called an ultra long read mode. ultra long reads being kind of loosely defined as reads that are beyond 50 kb. I think the record at the moment, at least when I wrote this, I think the record was 1.35 megabases, a single read that was more than 1 million bases long. I think this is actually out of date. I think we're actually closer to 2.5 megabases, something like that. But in fact, I think there is, I think there's a sort of contest between labs to see who can generate the absolute longest read. I have no idea who the owner of this trophy is at the moment. Okay, let's talk about some of the things that we can do with long read sequencing, and then I'm going to talk about the actual mechanics of doing those things, right? So I'm going to highlight a few different use cases. I'm going to highlight this one first because I happen to work on this project, but it's also a huge part of the Broad's long read sequencing portfolio at the moment, which and short read portfolio. So the All of Us research program. Which started in 2015, has released to date around 245,000 genomes sequenced with short read sequencing, on its way to 1 million genomes. Last year, we released the first 1,000 long read genomes. Later this year, I think we are on track to release somewhere between 5,000 and 8,000 additional genomes. And then in 2025, we'll release, I think the full set will be somewhere between 15,000 and 20,000 genomes. There's by the way, all of that data is publicly available on the All of Us Researcher Workbench if you're interested in going and playing with it. But I'll just highlight one of my favorite use cases from this. This is from Julie Wirtz, one of my colleagues at University of Washington, in Evan Eichler's lab. And what they've been doing is characterizing repeat expansions in a number of loci that have been that are known to be associated with genetic disease. This is from FMR1, the locus underlying Fragile X syndrome. You can see these interesting repeat motifs and the variation in the motif itself at this locus across a number of individuals. They're, interesting. The last 3 entries on here are individuals who seem to be at the pre-mutation length, right? So not pathogenic in these individuals yet, but certainly something that we would want if we were sequencing this person in a clinical capacity, might want to report back to them. All of Us at the moment doesn't have return of results for long read sequencing, only short read sequencing. I think this example is something that clearly sets the direction in which we're going, that eventually we would like to see the long read data in clinical practice to be able to report events like this, events that are completely invisible to short read sequencing, but quite well characterized with the long read data. What else? Let's talk about rare disease for just a moment. This is an analysis that was brought to us by our good colleague Anne O'Donnell, a patient with partial agenesis of the corpus callosum, on which short read exome sequencing had already been performed. And you can see in this plot there's evidence of 3 copy number variants, right, towards the end of chromosome 6, right? So two gains, which you see here, and 1 loss, which is really interesting. But short read exome sequencing being what it is, it's exceedingly unlikely that the exome data will have captured the exact break point of the event, which means that you don't actually know what the true nature of the event is. This is a sample that Anne and Elena England sent for long read sequencing, and then our team helped out with the analysis. And when we looked at this locus, I think this is work that Steve Huang on my team did just a couple of days in IGV, staring at these reads and following the breakpoint patterns until he could figure out what was going on. You can see these two copy number gains and losses are recapitulated in both the exome data and the long read data. But with the long read data, you now have this additional information of the exact breakpoints of the reads. And you can start to follow them around the genome. And so Steve reconstructed this locus and realized that it's not just two gains and a loss. It's actually a loss of section E, duplication of section A, and inversion of section C. Really, really interesting. What else can we do with these instruments, right? Like I said, the nanopore instrument is not an imaging-based platform. It's an electrical sensing platform. As a result, it comes in a variety of form factors, including these handheld form factors. I think this is the first time we've ever had a long-read sequencer that I can literally throw into a backpack and take anywhere in the world I want to go. And so this is an example where we did exactly that, although I'm showing well, you have to forgive me, the inset image, the plot on the right doesn't match the source of the data. The data on the left is actually from Senegal, and the data on the right is from East Africa. But the example here that I'm showing is the same, which is the increasing failure of rapid diagnostic tests to identify malaria in a patient. What's happening? Well, the rapid diagnostic test is kind of like a COVID test. It's antibody based. It's looking for two genes called HRP2 and HRP3, which we are seeing increasingly deleted in malaria samples. It's kind of crazy to me, but it makes perfect sense that that malaria is not only resistant to drugs and vaccines, and our immune system is developing resistance to the test itself. Because of course, if you were able to discover that a patient has malaria, you would treat it, which means that the parasites that don't have that locus will evade that test and continue to propagate. That's a deletion that we are seeing very cleanly in the long read data. But the interesting thing about this is where the sequencing was done, not in the Broad genomics platform. This was done on site in Senegal by Senegalese scientists. We're able to now take these sequencers anywhere we want and look at variation that we haven't been able to see before. Okay. Let's talk about analysis of the long read data. We've already talked about two of the pieces. We've talked about error correction. We've talked about methylation calling. I want to, those steps run on board the instrument itself. You don't have to do anything anymore. That's a fairly recent change. I think as these platforms mature, we are starting to see a pattern very similar to how I think Illumina sequencing progressed throughout the years. It used to be that you would get raw data off the sequencer in image format, and then you would need to run all of the downstream software yourself, right? The base caller that would look at the images and derive, the probability that some cluster was an A, C, G, or T at a certain time point. Eventually, Illumina moved all of that software on board their instrument, which, by the way, killed off a whole research arm of people trying to make better and better base callers. At some point, the only people who could work on that was Illumina engineers themselves. Same thing with PacBio platforms. Increasingly, a lot of the software is run directly on the instrument, so we don't need to worry about it anymore. That's true about error correction. That's true about methylation calling. It doesn't mean that we can't run our own tools, but it means that we need to take special considerations. It means that we need to configure those instruments to emit raw data that we can use ourselves, which is generally not default and generally not what we are doing for most of the projects. So all of that runs on board. Let's talk about the steps that don't run on board the instrument, the things that we are expected to do for ourselves. And there isn't time to give a totally comprehensive overview of the field. So I'm going to talk about the most common processing steps and decisions, the things that I think would enable, at least like 90 to 95% of, I think, the use cases that this audience would probably be interested in. The first thing I'm going to mention is the choice of the reference sequence, which matters very greatly. I think, you might be inclined to use the exact same reference sequence that you're using for short read data. In a lot of cases, that is the wrong thing to do. And it's kind of a mundane technical reason, but I think it's important enough that I've put it here. So the aligner for short read data is generally what we call alt-aware, in that it knows about alternate loci, alternate contigs for certain loci, right? So there are alternate MHC loci, and there are alternate loci for some other interesting diverse regions of the genome. Because the aligner knows about that, it knows how the genome the genome build is configured. It can still do the right thing in terms of mapping to the canonical representation and avoiding the alternate representation. But the long read aligners generally don't do that, right? They are not alt-aware, meaning that it matters if you are giving a build of the reference sequence that has a lot of redundant copies of a locus, because whoever prepared that build thought you might want to see the 6 or 7 different MHC copies and stuff like that. As a result, if you map to the wrong build, you get a pattern that looks like below, which is a whole bunch of reads that seem to not map properly to the genome, right? All of these sort of white ghosty reads that are mapping quality 0. None of these are aligning to the tumor necrosis factor gene, because there are multiple copies of that gene in the build for the reference sequence. So for long read data, it's imperative, if we're using HG38, that we're doing this with the version of the genome that does not have those alternate contigs and those decoys in it. This is something that our friends at, I think, Regeneron were messed up by a little bit. In the initial version of their exome processing, they had used the wrong build for the short read data and an aligner that was not aware of these contigs. And so there are a lot of medically relevant genes that ended up being mischaracterized in their first go simply because of the choice of the genome build, right? So before any of the informatics is understanding which genome to use. Of course, that picture is a little bit more complicated today. We now have, in addition to HG38, we also now have the telomere to telomere CHM13 reference, which has no gaps, where all of the chromosomes, including the acrocentric chromosomes, are 100% complete. Ribosomal arrays are present. What we've seen, again, this is a really nice result from Aganezov just a couple of years ago, is just the use of the CHM13 reference over HG38 reduces false negatives, dramatically reduces false positives, increases the discovery of SNPs and indels, increases the discovery of SVs. So without any changes to downstream software, just the use of a more complete genome gets you, unsurprisingly, a more comprehensive assessment of variation. Of course, in a lot of settings, I understand that this is not necessarily a practical move to make. We have this concern a lot for the All of Us project, where we have a huge number of clinical annotation databases that haven't been ported over to T2T yet, or other data sets that have been aligned to HG38 and not T2T. So it is a decision that needs to be made at analysis time. What is it that you are attempting to do? If you need to compare to other samples, that might dictate the choice of the reference build. But I'll tell you, for the All of Us long read sequencing, project, we've actually done both. We've aligned everything to HT38 and T2T to leave ourselves the option of being able to do this more this improved variant discovery, but not completely throw away all of the excellent annotation resources that we have in HT38, and also not alienate the clinical community that needs the data on one build versus another. After that is alignment. There are a few different aligners out there. We tend to use, we tend to see Minimap 2, which is a really nice aligner by Heng Li, who also wrote BWA, the short read aligner. This seems to be the one that is the most popular. And in fact, it works for both Nanopore data and PacBio data. PacBio, in fact, releases their own version of the aligner. They call it PBMM2. It is basically a wrapper for Minimap 2 that sets some additional options that are appropriate to PacBio data. After that is variant calling. Again, SNP and indel calling satisfied by a bunch of different algorithms. I think at the moment the most capable that we find is deep variant. Google and PacBio have been very strong collaborators in recent years. A lot of the onboard error correction software for PacBio data now comes from Google. So a lot of the machine learning infrastructure that is now being applied for error correction is not coming from PacBio themselves. It's coming from Google. Their training, their SNP and indel caller has this wonderful characteristic of being able to be trained on increasingly large data sets. Right? For people who are not familiar, deep variant is basically it's a variant calling tool that treats variant calling as an image recognition problem. So if you've ever had this feeling of, I don't know why the caller didn't see it, I can see the variant in IGV, it's quite literally the inspiration behind deep variant. The team that wrote this tool is very near and dear to my heart, because the person who led this team was my former boss at the Broad before he left. That works for both PacBio data and Nanopore data. A lot of the analysis tools are kind of interchangeable these days between two platforms. You don't really need to find a completely separate tool chain for PacBio data and for Nanopore data. I think, even though the platforms are quite different, I think the tool authors have been very mindful of making their algorithms work for both platforms. Structural variant calling as well. This is a place where we find that there are a lot of different choices, and it's the individual callers are still evolving very quickly, and they also have differential capabilities. Right. So the two that I've mentioned here, PBSV and Sniffles. PBSV is written by PacBio. Sniffles is written by my colleague Fritz at Baylor. But both of them are largely doing the same thing. They're relying on data that has been aligned to a reference sequence. They identify in the alignment pattern signatures that look like they are putative structural variants. They cluster those structural variant signals. They filter out some of the low quality reads. Summarize those reads into a consensus call, and then attempt to compute the genotype based on how many reads support one allele or the other. But increasingly, we see de novo assembly applied to these genomes in order to achieve much more comprehensive variant characterization. These are two callers. I apologize. The titles for the two callers fell off the screen. But on the left is Hi-Fi ASM, and on the right is Verco. Again, both of these algorithms these days work for PacBio data and Nanopore data, but interestingly, they can combine PacBio and Nanopore data. So combining the long read data from PacBio platforms, which tends to be more accurate, with ultra long reads from Nanopore in order to overcome some of the most intractable repeat regions in the genome. And last but not least, copy number variant calling. This is a wonderful tool by some of my colleagues in the data sciences platform, really designed initially for exome sequencing data. Very happily, it works on long read data as well. We've used this to great success. This example that I just showed about the patient with agenesis of the corpus callosum, the copy number variant calls here are made by GCNV. In our infectious disease work, we've been looking for amplification of certain genes that are responsible for drug resistance. This locus in sample DD2, you can see an amplification of MDR1. Again, nanopore data that we generated in the field, but GCNV is very capable machine learning algorithm that is learning patterns of coverage across the genome and is able to ascertain the presence of an abnormal excess or loss of coverage at a locus to make these calls. There's a wealth of tools I didn't go over. I wasn't able to go through methylation calling and some of the newer tools available for structural variant filtering and characterization. We'll have to do a second session in the future. But for now, I want to leave time for comments. Let me just thank all of the people on this slide. I am tremendously lucky to be representing the work of a huge number of people, and I thank them for their contributions and their analysis and their mentorship and guidance. And I thank all of you for listening. Thank you very much.
Thank you, Dr. Garimella, for an impressive talk that's impeccably timed. I think we do have time for one question if there was someone in the audience. Yeah. Here, if you're coming up to the microphone. Yeah. It automatically turns on.
Oh, okay, it's on. So I have a question about the CNV calls, and you mentioned there are like 2 duplication and then 1 deletion you saw there. So like in the long read data in IGV session, you see this like amazing like duplication, inversion, and deletion. Can those calls called by the SV caller you mentioned for long read? And like maybe save the tremendous time you spend on staring at the IGV session and figure out what are those things?
It is an excellent question. The answer is sort of, right? So the reason that we're using multiple callers is because what we've observed, and I think this was very formally stated in a paper from PacBio and some colleagues. If you look up Wenger et al. 2019, Wenger et al. Show very clearly that the 2 callers that I mentioned, PBSV and Sniffles, which are based on alignments of reads to a reference sequence, are surprisingly insensitive to really large structural variants, right? Their sweet spot is things that are below maybe 1,000 to 2,000 bases. This event is much larger than that. So the reason that we apply genome assembly is because we can get a much better characterization of this locus. But in this case, I think there's a third thing going on, which is the structural variant callers are largely looking at individual reads and looking at signals within that read that indicate the presence of a variant. What we're seeing here, I think, is much more easily characterized by coverage gains and losses. That doesn't really come through in the reference-based SV tools that we're looking at. It doesn't really come through, like if you're lucky enough to have enough coverage to assemble through the genome, I think you'll probably see that. But in this case, we didn't. So the application of a depth-based variant caller, GCNV, was really critical for us to figure out like where exactly we should be looking for as the boundaries of this event. This is why I think our structural variant calling pipeline is an ensemble of tools rather than just one.
Thank you.
You're welcome.
Great. Thank you so much again. We look forward to hearing all the future mysteries that you solve for us.
Thank you very much.